In this video, we're going to take a look at what happens when meiosis goes wrong, when there's an error in meiosis. That process is called non-disjunction. Non-disjunction is a failure of the chromosomes to separate correctly during meiosis. It can happen either in meiosis 1 or 2, and this results in the addition or deletion of chromosomes from a gamete. So they, instead of having 23 chromosomes, they might have 22 or 24 or even a different number than that. The only way you can determine that has happened for sure is by performing a karyotype, which is where you take a picture of the chromosomes in a cell. So this is what a karyotype looks like. And this is a karyotype for an individual that hasn't had non-disjunction occur. So what happens is you arrange the chromosomes by size and in pairs. And basically, the easiest way to do that is to find a cell that's undergoing metaphase, so that they're all nicely lined up. You can take a picture and then blow them up. And in the olden days, people would literally cut the chromosomes out and line them up and take a picture. In a typical human karyotype, there would be 23 pairs of chromosomes. So you wouldn't do this in the sex cell, you would actually do this in the body cell of the individual so that you could see all the pairs. The last two chromosomes don't have a number. So the first 22 are given a number. This is a little bit blurry, but they're arranged by size from biggest to smallest, from 1 to 22. And then the last two are the sex chromosomes, and they're not paired up. They could be. If you have two X chromosomes, you're genetically female. Your sex is genetically female, so you would have XX here and nothing in the Y position. This karyotype is for a genetically assigned male, so this is X and Y. So they're biologically male, their sex. They're called the sex chromosomes. XX will be female and XY would be male. I don't know why the words got cut off there. So there is a condition called trisomy. So that's when there's an extra chromosome somewhere. So the person would have 47 chromosomes, not 46. So this is a karyotype from a female with Down syndrome. So the notation would be 47, meaning that there's 47 chromosomes. XX, meaning the sex is female. And then plus 21 means that there's an extra 21st chromosome. So trisomy 21 is one way of naming this condition. Its common name is Down syndrome. So there's a third chromosome here. Most examples of trisomy do not lead to the individual being able to survive much long after birth. There's trisomy 18 and trisomy 13. Those individuals usually either die before birth or shortly after birth. But somebody with trisomy 21 can actually live a longer and more productive life. You probably know somebody with Down syndrome and would be able to confirm that. Monosomy is when there's a chromosome missing. And if you're missing a chromosome in the 22 somatic chromosomes, you wouldn't be able to survive. You have to have 2 of each of those chromosomes. The only way you can survive with monosomy is Turner syndrome, and that's when one of the X chromosomes is missing, or even the Y chromosome can be missing. The person just has a single X chromosome. So the way you notate that one is 45XO. There's a missing sex chromosome. So that person would appear female, but they would have some issues with their fertility. They wouldn't be able to have children themselves, and for some reason their stature is smaller as well. And there are some other health conditions, but a person could live a long life with Turner syndrome. And they could actually avoid some of the stunted growth that comes with the condition if they took human growth hormone or other medications to help them grow a bit larger than their smaller stature. there are some other non-disjunction examples with the sex chromosomes that can occur. There's one called XYY or XXY, so you end up with two X chromosomes and a Y. That's called Klinefelter syndrome, or XYY. You can be XXX. So when the non-disjunction happens in the sex chromosomes, the symptoms aren't as severe, and you may never know that you had any of those conditions unless you did a karyotype. So parents can undergo genetic testing for non-disjunction or other chromosomal abnormalities or other genetic conditions. You can undergo carrier testing. So that would be when you would have tests done to see if you're carrying the gene for a condition that might cause harm in the offspring. You can have blood tests. After you've conceived and you're pregnant, you can have ultrasound, which includes nuchal translucency screening. That is checking for whether or not the spinal cord and the brain have properly been encased in the spinal column and the skull. There are some conditions where, like spina bifida and anencephaly, that can be problematic, where the spinal cord in spina bifida doesn't close off properly, or the skull doesn't close around the brain properly for anencephaly. You can also perform a process called amniocentesis, which is when a needle is placed in and some of the amniotic fluid is taken off. Because the fetus is developing in the placenta and swimming around in the amniotic fluid, some of their cells would slough off, just like we have happen from our skin, and you could take some of those samples and test. And then another process is called chorionic villus sampling, where you take a small portion of the placenta and test it, because the placenta is both maternal tissue and fetal tissue. So those are all examples of genetic tests that could be done.